First of all, thank you, everyone, to come to this wonderful meeting. Especially thank you to Mohit and Shaon and ICTS for having me here. I actually have been spending a week as a Simons visitor at NCBS Theory, and been really nice talking to people and connecting. I also, when the idea of ICTS was discussed early on, I literally, when I first landed here, I was like, oh, it feels like walking at IIS, because I spent a lot of time at IIS in Princeton, and it just literally felt like just, very quiet and serene, and you can be in your deep thoughts and think about problems. So, I'm giving 2 back-to-back lectures, so hopefully the first one will be not too terrible that you will stay for the second one. The first one is mostly focused on teaching, so it'll be a little bit basic and going over very simple concepts of how to think about decision making in single cells. And although my talk says cancer cells, you can really conceptualize this in the context of any cell type. Cancer is a good system to study for theoreticians and computational biologists, because it really presents a striking dichotomy of to die or to live. The cancer cell either dies or lives, whether in response to therapies or just when it first starts to form cancer. And okay, so first things first, I thought I will, by the way, what I did was I had planned to give a talk on something else, and then yesterday I decided I'll add a lot of new things. So hopefully, this is still a little bit half-baked, but hopefully we'll collaborate together and learn more about it. So this is a collaborative lecture, so I really want participation from everyone. A lot of discussion, please ask questions. Let's have a lot of discussion, and no question is stupid. I also want to quickly get a sense of background of people in the class in this conference. So how many of you are undergraduate students? Raise your hand. All right, a few of them. How many of you are masters and PhD? Wow, most of you are in masters and PhD, and any postdocs here? A couple of postdocs as well. And just in terms of general background that you're in, how many of you are based in general biology? Yeah, engineering, yep, doesn't matter. All of them are like very fluid, right? As the department, there are no departments here as well. So whatever you identify generally as, sometimes it's hard to identify as well. I just want to get a sense of what is, maybe like more biology versus mathematics and physics. So about 1/3 in biology, how many in math and physics? Okay, so about 40%, 50%, and engineering. Okay, so it's a pretty good mix. That's great. So it's not just all biologists, so I hope that this lecture will cater to a lot of you. And so with this in mind, I will try to adjust my future slides accordingly. So I thought so my talk is split into 3 different parts. Part 1 is more generally talking about better experiments and modeling lead to stronger conclusions. So I looked at the conference name, and even though it says theoretical approaches, but at the end of the day, it's still cancer progression in treatment. I teach a class at Northwestern on carcinogenesis, and one of the lectures I spend most of the time on covering some of the aspects that this very nice commentary or perspective took. So I'm going to cover some of these basic ideas with you all. So when you are designing experiments or theory, you keep these in mind for better conclusions or stronger conclusions. All right, so first, the idea of reproducibility versus robustness. Does anyone want to tell me what is the difference between the two? Or what they mean to you? Yes.
Reproducibility means that if there is an experiment, I can do it at my lab or my particular experimental setup. Say if it is a computational biology experiment on GitHub, I can do it on my local machine without it failing. Or like we do get problems sometimes, like R breaks down or Python like doesn't give the required result. And robustness means that it is tightly packed in the sense like the conclusions being drawn both with the dry and wet labs meet each other. And that's why, so reproducibility just means that you can regenerate the results, and robustness means that you can tell a complete story with the whole data and experiments that you've done.
Okay, thank you. Anyone else? Okay, and then we'll move forward. Okay, so yeah, so I think both of you covered generally the same thing, but sometimes people confuse the two. So reproducibility or lack of reproducibility is the inability to reproduce the findings of a given published experiment under Austin's, you can't exactly have the same, condition. Being a theorist, you can never have the exact same situation, but generally similar situation, can you get the same answer? Uh, what are the reasons for lack of reproducibility? And the reason I have a star here is because some of the part of my talk will be focused on what does it mean for something to be lack of reproducible in my research talk later on. So I wanted to just quickly get a sense of when people think of why do people get lack of reproducibility. Like the easiest answer is, there was just fraudulent results, right? Someone just faked the data. Are there any other reasons that people could think of that you could get lack of reproducible results? Or that, yes. Yes, and
the model or the experiments are not very well explained in detail, so that you can't really, like, you don't know what exactly to reproduce.
yeah, so there could be many different reasons, right? Yes, one more.
Systems themselves are pretty complex, and thus, particularly like cancer systems or such, that many questions that we would try to answer, depending on the design of the experiment, might just make them irreproducible. And there has been a lot of claims, like a lot of studies that haven't been reproducible, like particularly drug target studies and so on and so forth. So the complexity of the system itself can lead to lack of reproducibility.
Okay, great answer. One more, last one. Oh, I thought you were saying. Okay, so I think a lot of this is covered. I think one of the major issues that sometimes, beyond the obvious, that something could not be reproducible is just sampling issue. If you have a system which displays rare behavior, then you may just miss it, and you will see that much of modern adaptation was understood by this very basic idea that Luria and Delbruck almost 70 years first thought of and embraced. So they embraced lack of reproducibility, which I'll come to later. And we already covered lack of robustness. I think this is the major point. So robustness always comes with, I think you mentioned this, it always comes with perturbation. You can only comment on robustness if you can perturb the system in various ways. And robustness as opposed to reproducibility, even though they both are linked in various ways, robustness is like, if a system is robust or your conclusions are robust, then it'll likely make predictions which are more useful than not. And it becomes particularly important in cancer where the system is highly heterogeneous, so finding things that work more broadly is better than not. Okay, so the next point I want to cover is causation versus correlation. So if you have A that correlates with B, there are many different possibilities that could happen. Can someone, does anyone want to take a shot? What are different possibilities? If you see A is correlating with B, what do you see? Yes.
A correlates with B means that as B changes, in whatever pattern B changes, A changes along the same way.
Yeah.
So it could be with completely irrelevant observations as well. That, yeah. Okay. Sorry.
Yeah, so what you can see is there are many different ways by which you can get the same answer, right? So you can get, if A causes B, you will get a correlation. If B causes A, you'll also get the same correlation. If something else causes A and B at the same time, you'll probably get the same correlation as well. And then there is a case by where A and B can be simultaneously upregulated or downregulated, or whatever your assay is, you can get the correlation in those cases as well. So it's very important to distinguish between correlation and causation, which leads me to the next concept that I want to quickly cover. And these are very basic pedagogical stuff. We'll quickly ramp up, but I added these few slides last night. Necessity versus sufficiency. So what is necessity? Necessity is if A is necessary for B implies that if not A, then not B. So that's necessity condition. Sufficiency is if A, then B. And one step towards causation, that's not necessarily always the case, but one step towards causation is finding necessary and sufficient condition, and how close the system is to be able to directly say that A actually influences B. So I'll give you some historical examples which have major implications in cancer treatment opportunities. So I remember reading about when the first time BRAF mutant melanoma was being thought to be developed, to have a drug being developed against the BRAF mutant melanoma. At many scientific meetings and board meetings, there was a strong argument that since BRAF mutant melanoma is present in cancerous and non-cancerous cells as well, there is no point in building a drug that will maybe inhibit the mutant melanomas. Now we know it works very well, with an asterisk because the resistance comes back. But earlier on, there was a lot of pushback towards it because people did not understand the concept of necessary and sufficiency. A better question to ask, perhaps in that case, would have been: is the activity of mutant melanoma necessary for melanoma maintenance? Not necessarily whether the mutant melanoma is doing the same job in benign or resistant tumors. Another thing that people think often about is if there are multiple driver mutations in one cancer, they must require multi-drug treatments, and that is not often true. Oftentimes, a single drug can take care of most of the tumor because it's a functional output that might be necessary for maintaining the tumor. Not always, but in some cases. So it's important to always critically think about what does it mean to be necessary and sufficient to treat a disease, in this case, cancer. Lastly, I think it's a very important point. So correlation plus plausibility is not causation, and I'll go over what it means. So to establish A and B, let's say A is a protein and B is a phenotype. So deleting protein A results in loss of phenotype B. So that's a necessity condition. Activating protein A always produces phenotype B. That's sufficiency condition. Do note that satisfying these two does not necessarily say that the effect of A on phenotype B is direct. It doesn't distinguish between the two. But what happens in biology or many biological experimental systems is we infer plausibility. Infer causality from biological plausibility. You can see I made it more recently. So I'll give you an example of a very famous enzyme that had that had been implicated in many different cancers, MELK enzyme. And in fact, there were many small molecules that were targeting MELK enzyme in clinical trials based on its anti-tumor activity. And several millions and billions of dollars were wasted on this clinical trial because people did not understand how plausibility is not causation. What recent studies showed was that actually the small molecule inhibitor that was targeting MELK was, although killing the tumor cells, it was not killing by inhibiting the MELK enzyme. And this group by Jason Shelcher at Cold Spring Harbor showed that even if you remove all of MELK enzyme in most cancer cells, the cells survive. So the drug was not really targeting MELK. So it's very important to build careful experiments, careful computational frameworks that can really get at the question of what exactly is doing what. Okay, one more concept. Yes.
Arrive at causality from this thing, because even if you design a very intricate set of experiments, especially in a biological system, there are so many different other things which can happen or which can influence. Is it even possible to?
Yes, so great question, right? So causality in itself is a very philosophical concept, right? Someone will say nothing can be truly causal because we will always have like some variable we cannot control. I think it's more about, can you almost like in mathematical asymptotically reach causality. So as close you can be to understanding of the system mechanistically, I think better it is for the development of cancer. But you're absolutely right, because if there are too many components, you can't control what is leading to what, right? Okay, another interesting point that I had not appreciated much before, but after reading this paper, this perspective I really enjoyed and I thought I'll share with students is experimental assays or computational assays that rely on upregulation or downregulation of certain things. So either it's more of something or less of something. And it's remarkable that, in just a natural consequence of the system, that upregulation assays or increase in signal is much easier seen than downregulation of the signal, right? Because of better signal to noise, and there's much more, more places to go than not. The problem with false positives, more interesting. So there are also fewer false positives, right? As you can imagine, because there are many more ways to make something go bad than not. So it's just also a property of complex system. an aeroplane, right? Hopefully there are redundant mechanisms or robustness. But if you systematically remove components, it's not like suddenly the aeroplane or the flight will get better. It usually goes in the negative direction. So whatever you find in up assays are usually going to be more reliable than in down assays, because in down assays, so thankfully up assays are more informative than down assays. That is, there are many more ways to decrease the performance of a complex system than to enhance it. So it's good that we can detect up assays more. Unfortunately, in biological systems, experimental biology, we rely a lot on down assays. And that's a big problem, because you can just get things which are uninteresting and not useful. Take any level of biological studies that are done in cancer, you often rely on down assays, which will reveal things that do affect the system, but they may not be as important, because it's too easy to make something less or worse off than better off. So that's why the success of a lot of these preclinical studies, basic studies, do not translate to clinic, because it just fails in other ways. On target versus off target effects. So this is another thing that even both designing computational and experimental frameworks always think about: how does your molecule affect the downstream phenotype? And there are many different options, right? So one is you have an unintended target, an intended target, which so if you have a perturbant, which could be your drug, it acts on the target, which then inhibits the phenotype, right? Off target could be the perturbant acts both on target and unintended target, but the unintended target is what is doing the job. The example of MELK that I showed you earlier, right? But you would not be able to distinguish between the two if you didn't go a step further and did some validation and other things, which I'll come to a little bit. Then there is on target and off target, where both of them, when both inhibited at the same time, will result in inhibition of a phenotype. Or the fourth case could be either of them being disrupted can lead to inhibition of the phenotype. And it's important to distinguish between these possibilities when thinking about your system and experimental and computational frameworks. So one way to address this is doing rescue experiments. And this is not just, when I say experiments, I mean you can play around on your computer as well, because experiments in the end is just thinking about how do you rule out various possibilities. So how do you get rid of on and off target problem? One way to do it is if you can somehow make the drug completely resistant to the other possibility, you can potentially see if you get the same phenotypic outcome or not. The other way people do it is epistasis, where if you know that one or two things are related to each other, or A affects B affects C, then you try to restore the downstream function by adding something in between, such that it rationally makes sense. Okay, so this is all this thing that I added recently, but as young computational biologists or biologists going into cancer biology, it's very important to deeply think about your work and how you can eliminate sloppy conclusions by being more rigorous about your experimental and computational framework. Yes.
Sorry, to be robust, does it mean that how good can a particular model answer number of questions, right? Does robustness means that how many questions does a model answer?
It's not necessarily about model, but have conclusions or experimental systems that are not very specific to a specific model that I guess in a modeling sense, you could have the model to be robust to different types of parameters in some sense, right? Or an experimental system, if you want to say this drug inhibits something specific, then you want to test it in many different cases. Another thing that this perspective argued, which I really liked, was it argued that in future in cancer biology, we should move more towards less number of conclusions, but solid backing of those conclusions for a paper to be considered acceptable, than many conclusions but not backed properly. So it's kind of coming to the same point, that there should be many different ways by which you can make the same point rather than make many points in one paper.
Say, if you want some kind of parameter to have robust enough studies, like some kind of confidence interval or p-value to support your claims or whatever experiments that you're doing, so that you can have some kind of parameter to say this is robust enough, like confidence interval of 95% or something along these lines.
Yes, I think you can have statistical significance. I think what I'm trying to say is a little bit more beyond, a more general, conclusion about how, or like statement about how to design experiments or computational framework is to make ones which are more general to the extent possible. Of course, you can't make something super general, right? Then you don't really learn much about the system. But make something, that is able to. It's not just about statistical significance, to say it simply, but I think a lot deeper than that in terms of how you think about your question and what it means to reach a conclusion in that question. Is it just like associated with or linked with, or is it much more than that? In fact, I would suggest to young scientists that just stop using the word associated with or correlated within your work. I think that will make you smarter and better, more rigorous scientists. Whenever you have to say something is associated with, I think that means you don't really have a lot of understanding of the system. That's my view on this. So we will take a 5 to 10 minute break, but I want everyone to think about what does mechanism mean to you. I say this because as someone who's not biology as an undergrad and go to a lot of biology talks, how do people think of mechanism? And I want to just highlight some of the ways by which you can think about what mechanism even means. So maybe we'll take a 5 to 10 minute break. And each of you, please think about what mechanism means to you, and we will just go over and discuss some of this in 5 to 10 minutes. So we'll end this session at 11:10 instead of 11, and we'll just use this 10 minutes to think about it. All right, let's get started. So who wants to? Okay, so who wants to go first in telling me what does mechanism mean to you? There's no wrong answers because most of us don't understand what it means, and it depends on the context, but I'm really curious what people think. Yes. No, there is an extra mic here, which I don't think these guys will use.
So I thought that mechanism is assuming that any system that we study is like a large machine.
Go ahead. Keep talking.
Large machine. So yeah, assuming that cancer, like when we're studying cancer, the system is a large machine. It is made of intrinsic parts, which may or may not be interrelated or with each other, and or might be connected with each other, influence each other. And this is exactly to figure out how that system is or what that machine is the study of mechanism.
So if you have a machine, just to paraphrase, you're saying if you have a machine and it has parts, and how those parts work together for the machine to function, that's mechanism. Okay. More? Yes.
In your phenotypes.
So set of processes, sort of related to this, set of processes that, did you say interact?
Outcome, it will give some outcome, and the
Results
outcome.
to an output.
Yeah. And it will have some impact in the system, like if we're talking our cancer biology, or that's.
Okay, so that's mechanism. Great. Yes? Maybe say your name and then.
I'm Adrian from NCBS. So it's all relative to two events, one at the beginning and one at the end. So you find a set of necessary steps that connect these two events. So any such time-ordered necessary steps would constitute a mechanism.
Nice, very nice. So we are getting into nuance of things. So start and end, and all that matters is how do you go, I guess, steps in between or something. Yeah. Okay, so kind of here, like interaction between parts. Yeah. Interactions. Okay. What kind of interactions? What does interaction mean to you? Like physical, or it could be indirect? So they have to like physically interact with each other. Okay. Does it need to be physical?
No.
Okay.
Generation of positive influence and negative influence by generation of signaling pathways between the cancer, and immune evasion is also a mechanism in cancer's biology.
Okay. So a signaling pathway is one example of these steps. Yeah, and you can go higher or lower.
Like PI3K AKT pathway is resulting into the proliferation of cancer cells.
Yeah, so some input leads to some output.
Yes, sir.
Yeah.
Like positive influence and negative influence on
Sure.
the cells.
Okay. Louder. You say your name and then everyone say your name and then talk.
Hi, I am Teja. So mechanism for me is like understanding the regulatory network that is governing a given process.
So there are different applications of mechanisms. So some people are interested in studying at the signaling level, some people at the gene regulatory level, right? Example applications or model system could be signaling, could be gene regulatory networks, regulation. Anything else? Not applications, but more about what mechanism means to you. Any more? What are the characteristics of mechanism?
Okay, I'll take the bait.
Like for example, you go to a talk and a biologist say, where is the mechanism, right? So what are they trying to say, or what do you think they're trying to say?
Hi, I'm Mahesh, and what mechanism means to me as a bioengineer or biologist is how does cancer spread, in like what factors do lead to that. For example, transcription factors may be upregulated, or some genes might be downregulated to, or some proteins are overexpressed or underexpressed, depending on different conditions and what we are trying to study. Say invasiveness, if we are trying to study how fast does a cancer spread, then we might be looking at different mechanisms, maybe like different transcription factors which are activated in a particular case, and which pathways are they using, say, and say like using the consensus DB or like any other pathway analysis we can do. And what other things that I think mechanism really means in a cancer biology context is if we can also have some kind of statistics or some kind of math behind or formulas behind the level of activity of a particular, say, RNA expression or transcription factor activity. So in that case, mechanism would be a lot more meaningful to me.
So if you can model something with mathematics or statistics, yeah. Okay. Mechanism more meaningful. Okay, last couple of answers. So yes, go ahead. Go ahead, yeah.
Yeah, I'm Srishti, and I think it's kind of related to what has been said. But for me, mechanism is very related to causation, or what causes what I'm studying. So that is an important aspect. If I know whatever I'm using to model my system, it's causing the result, then that is.
So how would you test it? So good. So mechanism is always tied with the perturbation or something, right? You can't have mechanism if you don't know how the system behaves when it doesn't perturb. So to get to causation, maybe what you're trying to say is that you need to perturb the system.
Yeah, just to add to that answer, yeah. It could be in part or in terms of complete. So a mechanism slightly varies from causation in the sense that probably the contribution of that mechanism to that particular process need not be the whole story. There can be other aspects.
It only tells you in the context, in that specific context. Great. Okay, so let's move on. Maybe last. Yes.
Pattern between the direct and indirect variables. Like everyone said, ki gene expression can be correlated directly to the endpoints. If we are studying two genes, there can be different paths which it is following, or it may be directly interacting with the two endpoints. So the pattern of the direct and indirect variables.
So like trajectories leading to endpoints, right? Or paths to the endpoint. Okay, so what I did was I first asked ChatGPT, because why not, right? This is the GPT 4.0, so it's the latest. I did it yesterday. So what does it say? In biology, a mechanism refers to a series of events or steps that are involved in a particular biological process or function. Kind of tautological, right? But whatever. Mechanisms are used to explain how biological systems work, and they're often used to describe the underlying cause of biological phenomena. A lot of these things are encompassed in this little definition. But I think they had a couple of examples in between, and then At the end, they said, understanding the mechanisms of biological processes, an important part of biology, as it allows scientists to develop hypotheses, make predictions. So no one talked about predictions, right? So it's one thing to recapitulate with model or mathematics what is happening in the system, but I think it's not very useful if you can't make useful predictions, right? Oops. Design experiments to test the hypothesis, right? I think this is not a definition of mechanism, but it's a consequence of mechanism or the use of mechanism, right? In many cases, finding mechanisms may still not be enough. So let me give you an example of if a biologist were to find a mechanism of how an airplane flies. How many of you have seen a Western blot before? A Western blot? Since not many have seen, I will first explain that before we go to the next point. So Western blot is one of the oldest biochemical techniques where you are able to see different protein amounts if you run it on a gel. Sorry, yes? What? Okay, yeah, so most people don't know. I'll just quickly explain what a Western blot looks like. So Western blot is like a 2D gel on which you start with individual wells in which you put in your protein of interest, or not protein, but like cells of interest, whatever you want. And then with because of some electromagnetic or like electrophoresis, you start a current from top to bottom, and your protein flows at different pace. Some can go very fast, some can go a little slow, and so the larger proteins will go down slower because of its mass, right, and other things, and the smaller proteins will go faster. And then you create an antibody, or you have an antibody or something that binds to your protein of interest. So at the end of an experiment, you get a plot that looks like this. So pretty much every biology paper would have Western blots until recently, and even now it's used very widely. So let's say you want to look for your signal. Your signal is signal A. So if you want to, you design an antibody or a reagent that binds to your signal A. So let's say you have sample 1, sample 2, sample 3, sample 4, sample 5, and your signal is present. So the darker the band is, where your reagent binds to, the stronger the signal is. So how I would interpret this result would be that sample 1 had a lot of my signal A, sample 4 had the most, and 3 and 5 had nothing. This is like the mainstay of biology, and several Nobel Prizes have come out of using this simple assay. If a biologist were to make a diagram of how an aeroplane flies, this is how it will look like. So this is how would we check with a Western blot successful flight activity? In wild type or normal case, you will have a lot of signal. When there is no pilot, there will be no signal. When there is no wing, there is no signal. When there is no passengers, less problems. So very high likelihood of better flight experience. And if there are birds, all kind of things. So this is the current state of biological mechanism, right? So we are so far away from understanding how biology works. In fact, we are just starting to understand biology. And what mechanism means is very much dependent on the context and the system. And this, in many cases, would be enough to say that I understand how an aeroplane works. But really it doesn't, right? So as young students who are getting into biology or thinking about modeling in biology, it is very important to push the boundaries of what mechanism means. Do not stop at very simple understanding of how things are working in biological systems. But this is truly the state of the art. You can change it to fancier methods, but it's still the same. We really lack a better understanding of how systems work. Okay, from this, let's move on to my actual talk, which I prepared for this session. How much time do I have? 40 minutes. That's great. So this will be more technical, a little bit more technical, and hopefully you will take some messages away. And I'm happy to talk about mechanism and other things, at lunch or in the evening or whatever. So this was the title of my talk. Well, it's a little modified, but how do cancer cells make decisions in the face of selective pressures? The selective pressure could be that of a drug, could be its microenvironment, could be other competition of cells nearby. I'm trying to keep it a little bit more vague. So over the years, pharmacological therapies have become very, very effective in treating cancer. So if you look at just the history of it, some of actually radiotherapy soon after X-rays were discovered by William Röntgen in 1980s or 1990s. was first used to think about treating cancer by introducing mutations. And about 50 years later, chemotherapy came into picture. Actually, people don't know that immunotherapy came before targeted therapies in late 90s, but now because of various synthetic biology approaches, immunotherapies are becoming very popular and very effective. And more recently, in early 2000s, targeted therapies came into picture where instead of chemotherapies, which broadly inhibit processes in a cell, targeted therapies are designed to specifically target a molecule, or at least that's the plan. And they've become very popular. Unfortunately, cells are very smart, and even after the success of these therapies, the problem remains. So this is a very classic example of a person with metastatic melanoma. After 3 months of targeted therapy treatment with vimrafenib, the patient is virtually tumor-free. However, 6 months later, the same patient, the tumor comes back, and notice that it comes back in the same locations, right? So what might be going on? And this leads to the question of what makes responses to stress different for some cells as compared to others. So clearly, there's something that happened at this in-between stage where for various reasons, the tumor came back after the therapy was removed, right? So how did people start thinking about this process? Forget about cancer for a second, right? How are people thinking about what makes some cells respond differently than others? You can go all the way back to Mendel and Darwin, where they talked about Darwinian selection, where if you have an original group of giraffe and some are taller than others, natural selection will favor longer necks. And I'll connect it to the cells in a little bit, but so over time, after many generations, the group will still remain variable, but it will show a general increase in neck length for giraffes. The competing hypothesis was Lamarckian, where Lamarck argued that over time, because to reach higher trees and things, giraffe will have to stretch its neck, and over time the neck will be stretched such that progenies will have taller neck as well. So it's really driven by the environment more so than selection of random states beforehand. We now know that the Darwinian selection is what happens in most cases, if not all, and that's well-established fact, but back then it was not well known. But let's go back to the scenario of cells under selective pressures, because going from species to cells, our resolution of techniques have improved, and we know a lot better, and we can look at more deeper, at higher resolution of biological scales. So the first answer to this question was provided by Lurie and Delbruck in 1943. I actually think this is my favorite experiment of all time. I think this was probably 30-40 years ahead of its time, and a single experiment got them Nobel Prize, both of them, right? So what do they say? This is coming to the reproducibility aspect I mentioned at the start of my talk. So what do they say? It is the belief of the authors that the quantitative study of bacterial variation, which until now has made such little progress, has been hampered by the apparent lack of reproducibility of results, which, as we shall show, lies in the very nature of the problem and is an essential element for its analysis. So what was happening at this time? There were two different groups of people. Some were saying it's Lamarckian, some were saying it's Darwinian, but the bigger problem was the results were not replicated in different laboratories. And because of that, people were not interested in solving this problem. But they embraced this lack of reproducibility, and they wanted to know exactly why it is not reproducible, right? So with very cool and smart experimental setups, and I will just very quickly give you the gist of it, but I recommend everyone to read this paper because I think it is one of the most beautiful theory and experiment paper together ever done. So what do they do? So they argued that, so there was no, this is time when very little about cells. There was no molecular biology, right? This is all 1940s during war, actually. So what they did was they argued that if you start from a single cell or a group of cells and you let them expand, and then you give them drug. So they were studying this in the context of a bacteria being attacked by a phage virus, right? So think of it as a host and stimulus, or host and stress. In this case, the host is the cancer cell or bacterial cell, and the stimulus is the phage virus. In cancer, you can change it to host is the cancer cell, and the stimulus or stress is the drug you are interested in studying, right? So they said that if I do these experiments many times, starting from a single cell and then give them drug, or in this case, phage virus, if the effect was a result of of mutations that happened before you gave the phage virus, or things that changed before you gave the phage virus, you will get what is called as jackpots, such that in many of the plates at the end of the experiment, you will get no bacteria surviving because most of them will die when exposed to phage virus. But in some cases, you will get jackpot plates. They compare it to slot machines because it's a very rare behavior. So what you would see is that in some cases, you will get very high number of bacteria surviving, and that possibly could explain that it's because of things that happened before you added drug or added virus. So they add virus after they let them divide for some time, right, at this stage. The alternative was that it is the effect of the stress of the phage that introduced various mutations or changes in the bacteria that allowed it to survive. In that case, you will expect the distribution to be more Poissonian, such that some will have more, some will have one, so it will be like a nice distribution. So in 1940, they just worked together and they showed that in their actual experiment, they don't have a plot for it. In those days, it was just tables and interpreting things. So they show that in most cases, the number of bacterial colonies that survive is basically 0 in most cases, except for some jackpot plates. And they use the simple idea of variance and mean to get at this question. So they argued that for a Poisson distribution, the variance and the mean will be same, whereas for something that is very heavy tail or much more weird things are happening, you will get much higher variance than the mean. And they show very conclusively that, at least in their system that they studied, and now many people have shown this to be true in other cases, that it is the pre-existing mutations that can lead to diverse behaviors consistent with Darwinian view of selection. So this was the first time someone did experiments, no molecular biology, don't know the structure of DNA, but they could make conclusions which resulted in Nobel Prize, right? So the model that has come to be appreciated in not just antibiotic resistance or other diseases, but cancer as well, is that there are pre-existing extra mutations within cells, which when you respond to stress, some of these extra mutations may enable them higher fitness, which can then lead to proliferation of these cells, right? So I'll just, before I went further, I thought, since this is a lot of students and I want you to be more inspired by this experiment, because this is truly my favorite experiment of all time, or quantitative experiment of all time. So 1940s, right? Nowadays we talk about interdisciplinarity. It was not really new, right? People in 1940s, physicist Max Drillbrook and microbiologist Luria, they were talking with each other, not for any checklists or saying that we have a biologist and an expert and a computational or a physicist, but really trying to solve big problems in biology, right? And second, they embraced uncertainty and lack of reproducibility across scales, which we briefly covered. So as young students, it is important that if something is not reproducible, it is often a good idea to really try to get to the bottom of it, because who knows, you'll find something very cool. Lastly, the structure of DNA was not even known back then. So 1940s is when they did their experiment. They had no idea how DNA looks like, but they could make conclusions which stood the test of time, right? So that makes this study even a lot more powerful and a lot more something to be desired, right? So this is the model that have come to be appreciated in cancer biology. Since 1990s, we can sequence a lot. We sequence a lot at the end, and we try to infer if some of those mutations were present before or not, right? So presumably this person is, or seemingly tumor-free, but there are very rare cells that have survived at each of these locations, which can then repopulate the tumor. Any questions so far? Okay, so we'll move forward. So I thought, let's see if this, what other places have people used this beautiful framework to get at important biological conclusions in cancer biology. So look at some of the papers historically. The same question can be asked not just about resistance to stress, but in metastasis as well. So there's a very classic paper that asked the same question in metastasis, where what they did was they started with this melanoma cell line, and they injected half of it well-mixed solution in a mouse, and there, since the mutations have already happened, you would expect a more like a Gaussian or a Poisson distribution. But if you make individual clones of it, like Luria and Delbruck did, if it was pre-existing variability in the primary tumor, you would expect that some of the metastasis to lungs will be much more in some mice than other mice. And they exactly showed that this is the case, because at that time there were competing hypotheses that is metastasis driven by signal from the distal sites, or is it driven by pre-existing differences that drive these cells to these distant sites? And again, it may not be a general conclusion, but largely, it's very well known that a lot of differences, whether it's genetic or not, that's a different question. But pre-existing differences can drive behaviors in cells and make them be more metastatic or less metastatic. Like work of Herbie Levine and Mohit here have shown a lot of EMT-driven states. So this paper does not really measure the mutations. They just say there are differences that exist beforehand which can drive the behavior. And at that time, most of the people thought about mutations. So we'll come to how these behaviors can be driven by without mutations as well. But the point is, there could be pre-existing differences which could be amplified later or not, but they drive a lot of behavior. So selection is taking place even in metastasis, for example. Then the question that comes to mind is, like even for Lurie and Delbruck, even though they estimated mutation rate, they never showed that it's mutations, right? So can we confidently say that it is DNA mutations that are the sole drivers of differential response to stress? Do we have concrete evidence that that is the case? In fact, if you look at human body, you have probably have a lot of mutations, but many of your cell types are not driven by mutations. So it's not inconceivable to think that things outside of DNA mutations can lead to interesting behaviors. So this leads me to conceptual point 2. Can non-genetic differences also result in different response to stress? This a lot of commonly used these days in presentations where people talk about non-genetic variability. It is now very well established by various studies that you do not need mutations necessarily to drive interesting behavior in cancer, and it was recently included in the work that this is a recurring set of
hallmarks of cancer paper, right?
Yes, so there are many. So every few years there are hallmarks of cancer papers that come out, and the most recent one included phenotypic plasticity or non-mutational reprogramming as a way by which cancer cells become a response to stresses. Could be of resistance, or could be metastasis, could be tumorigenesis. But then the question is, when and how does it act, right? And so this is fairly new, recent field. Single cell technologies have enabled us to really profile things in various ways. And when I say single cell technologies, I don't just mean sequencing, which is how many people think, but single cell biology is a much deeper or more conceptual field than not. But newer technologies are allowing us to really profile things at single cell level. Yes.
Like one clarification, trying to understand, like you told that if we consider the Darwin selection, like the height of the giraffe is getting higher because DL, both the hypothesis, right? It is telling that survival for the fittest because it has to reach certain height or something. So like, and you try to connect like when you are giving therapy and after that the tumor is coming back after a certain time, and then you're telling that the mutation because some cell has the more prone to mutation, so there is chances that they will come back, and then you try to connect this like how?
Yeah, so what is your protagonist in the survival of the fittest, right? In some cases it's obvious. In this case it's the cancer cells, right? So presumably it's hard to say that that there is some brain behind it, but the cells are trying to survive, and there are many ways by which they can survive, and some cells have competitive advantages and they repopulate the tumor. When you think of evolution, you always think at much longer time scales, like what it means in terms of cells coming back and becoming resistant. They want to live, right? Whether that is good for Earth, that's a completely different question and philosophical question, and it's much longer time scales. But at just like from observation, these cells want to live, right? There are ways by which they want to continue growing and spreading their population. So it's very intimately linked. But whether calling one is advantageous or not, advantageous to what, right? It's always relative to what. And as a system, you can't predict how individual things will contribute to the survival of the fittest longer term.
So the driver can be many things for metastasis or cancer relapse, right?
Yes. So mutations, non-mutations, and I'm coming to that.
Yeah.
Any other questions? Okay, so we know that a lot of this is driven by non-genetic behaviors, and you don't need mutations for exciting and interesting properties that happen in cells, especially in cancer cells. But how do we get to the questions of when is this happening and how does it act? And at least there's a huge field, and people are thinking about this at many different time scales. But at one time scale and one aspect of things that our lab is interested in, but I'm not going to talk too much about our work here. That's for the next talk. Can we find structure to this variability, and what kind of variability it is that matters? First of all, even going beyond correlation to as close to causation as possible, how do you go about doing it, right? So how do we go about testing the contributions of non-genetic differences in cancer and metastasis? Again, Luria-Delbruck analysis to the rescue. So this is from a very beautiful paper by Sidney Schaffer, who showed that. So this is the original Luria-Delbruck test, which I just showed you before, but in a slightly different form, where you start from a single cell and then you let it grow for a long time, and then you add drug, right? So you will get jackpot plates, where some plates will have a lot of resistant cells and some other plates will have nothing. So the distribution will look something like this, where most of them are 0 and then there is heavy tails. So the variance is much higher than the mean. But if it is non-genetic, what happens? In non-genetic, it is possible that cells are transitioning between different subtypes, where some are a cell that is in this green abstract subtype state is the one that is going to become resistant and repopulate the tumor. So if you were to do the same experiment that Luria-Delbruck did, you will see that it'll be, since cells are transitioning back and forth, and if you know the general time scales, you can design the same Luria-Delbruck experiment, and maybe you will not see a distribution that is heavy tail, which is exactly what they showed. They showed that in this very highly controlled experimental system, you can get distributions which are not heavy tail, but it doesn't rule out the fact that it was pre-existing. So heavy tail distributions work more clearly when it's a permanent change. When it's a non-genetic change, then you can get a rich set of outcomes. And this is not just one. They did many replicates on many different clones, many different types of melanoma cell lines, and they always see this. It's really beautiful experiment. For the first time, people used this kind of analysis to look at non-genetic variability, at least to my knowledge. So you can use Luria-Delbruck to get at the questions of the time of how you can look at non-genetic variability and potentially quantify its time scales. Yes. Extremely important, right? So if the number of generations are shorter than the time scales, then That's a great question. Actually, I would say that in this field of cell state transition, finding timescales is one of the biggest open questions, and what leads cells into that state and get out of it. It's also hard because there's no in mathematical terms, like two fixed points, right? It's a lot in between. So how long do cells take to go in and out is an open question. In melanoma, they could get at this. So I think they went a little bit more blindly into the problem. Yes.
Talking about non-genetic mutations between ones that are induced and not induced? Because both of them will give you a non-flat tail distribution.
Yes, that is a great question. So I think the reason they could conclude that is the timescales were very short. So if the timescales were very long, so these experiments were done on the scale of 2 to 3 weeks, and I think they did some whole genome sequencing at the end to show that there were no acquired mutations during the process, at least in that paper, when they have it in.
It's not even acquired mutation. The acquired phenotypic change that wasn't there previously, that was induced by the.
Oh, so that does happen. So
that does happen. So how do you tell the difference between those pre-existing and not pre-existing?
So I will come to that later. You can do barcoding to do those, but at this stage you cannot distinguish between whether it was pre-existing and that gave them some advantage to reprogram later, or it was completely driven by induced reprogramming. You're absolutely right. Yeah. But timescales is very important, right? So now people are slowly trying to put a number to it, but at that time they didn't know. They just really went blindly into it. I do not know if it is linked to mutation time scale. The more it is in drug, the higher the chance of getting new mutations, right? Because there's more selective pressure of the drug. But I don't think at short time scales that will matter. It can induce phenotypic changes, permanent or sort of permanent phenotypic changes. But at these time scales, you don't get mutations. What can happen is, and we have seen this in some of the cases in my own system, is if you keep them for very, very long time, you suddenly get a mutation that exposes those cells to a new set of substrates, right? So you will get mutations which will lead to the cell having access to a lot more cell states which were not possible before. So also like finding the time scale of interactions and which time scales non-genetic versus genetic works, that's also an open question in the field, and many people are interested in it.
One quick question, and following up on Mohit's question here, I mean, if you see that the mean is not equal to the variables, you already know that there must exist some cell states or something before the drug was added, right? So you may not be able to distinguish which of these states came up after or before the drug.
Yeah.
But you can at least argue that some of them must have existed before.
Yes, so you can decouple. Sometimes you are not very powered experimentally to have a distribution which, so you really need smoking guns difference. You really need like strong variation differences. In this system, we do know conclusively the answer is that there are pre-existing cells which are slightly different, but when you add drug, they reach a new phenotypic state which is very stable. So when you remove the drug, they don't go back, but there are no mutations. So it's almost like there are pre-existing differences that give you a kickstart. And once you have that kickstart, maybe I'll leave that for my research talk. So we'll go over it later. It's more than Poisson, of course, yeah, but not that high, right? That you would expect for a 0-0, of course. Yes, very true. Of course, it's 10 times. Yeah, so it's not Poissonian, but it's not as extreme as you would see for very jackpot cells, where most cells are 0, right? Okay, the third is, can we measure the time scales of variability that matters? So we know that there are non-genetic differences that can lead to different phenotypic outcomes. But can we also use the same, it's like one-stop shop for everything. We are using it for immune cell responses in our lab as well, but can we use Lurie-Delbruck fluctuation analysis to measure the timescales of variability that matters? So what do I mean by that? So you can get cases where there are various expression programs or gene expression programs that when cells divide, let's say you start with a population of cells, and there are some cells which have certain gene expression programs or transient phenotypes. After a few cell divisions, it is possible that these variabilities were operational at timescales closer to the cell division timescale. So you will get a distribution such as this, where a couple of cells in a growing clone will have this transient cell state. The other possibility within this spectrum is that you have heritable expression states, where when you let the cells divide, these gene expression states persist for a long time, but they're not necessarily permanent. How do you distinguish between these two variabilities? So again, the same person, it was actually this person's PhD for 4 years, a Cell paper and a Nature paper in 4 years of PhD, really major contributions to single cell biology. But how do you differentiate with Lurie-Delbruck kind of experiments? So this, you can basically do the same experiment without introducing any stress or a drug. You can just start from single cell, let them divide and grow for a few generations. And if the state is heritable, you can see that some genes, they turn off, so it's not permanent. And you do, you collect many of these clones and you do gene expression, RNA-seq or something else. So it's also a very cheap experiment. It's not single cell, right? What you can find is you'll probably get, histogram of gene expression across 48 clones to be more like a bell-shaped distribution. Whereas for heritable states, you can get distributions where there's a lot of variance between the population. And I'm not going to go into details of this, but feel free to read this paper. But by using this approach, they were able to find genes which have a memory, they call them memory genes. Genes will persist for a long time. So it's heritable, but it's not permanent. And these genes tend to have a lot more, these genes tend to have functional consequences with respect to drug treatment, but also more recently, there were some papers from Mark Dawson and other groups showing that these kind of, and Tyler Jack's group, that some of these heritable fluctuations also are implicated in metastasis and clonal fitness across tumorigenesis as well.
Sir, are you trying to say that these memory genes are also a lot more conservative as generations are, as we study them across generations, across time?
Sorry, say again.
These memory genes that you recently talked about, are they a lot more conservative, or are they conserved across generations, something like that?
Yeah, they stay on for a longer time than cell division time. Yes, so it's not just random noise. There is some regulation behind it. Okay, last point, and then we'll end this. How much time do I have? Okay, so great. So we'll finish on time. So the last point I want to make, and I think it's very important for young biologists and computational biologists, is that the eventual fate of a cell is not necessarily predictive of the driver state. And I'll unpack this a little bit. So in some cases, we can more, so in cancer biology, we are often limited by looking at endpoint. After the cancer has happened, we try to infer what drove it from mutational profile. After the resistance has happened, we profile the cells and we try to understand what may have driven it. But eventual fate of cells is not necessarily predictive of the driver state. So let me give you an example. So this is me last year, and this is me in 1998, about 7 years after I was born. And you can generally see, okay, this is probably Yogesh, 20 years later or something. But in many cases, we cannot infer. So when can we infer, right? When the fate is simply a selection of a state, right? So a very famous Hollywood actor, Paul Rudd, who's at 35 and 52 looks the same, Rekha in Bollywood has the same. So if it's such a selection of state, you can give older pictures and say, who is connected to who, like can you build a connection between a state of a cell and final fate? You can do it confidently. When the time scales of separations are not very long. So if you're given these 3 pictures and you were to predict who is this, I'm pretty sure you can't predict that this becomes this person, right? So the closer you are in the time scales to the endpoint, the higher the likelihood that you can predict which cell state drove the process. Lastly, there are also cases where the alternatives of the state before, because oftentimes in experiments, we only have the current endpoint, right? And we are trying to use that to infer what happened before. But if the possibilities of states before is too variable, or not variable, too extreme, then it's also easier to predict. So if you were given 2 babies, a white baby and a brown baby, you can probably say this is Yogesh in the future, right? So also think of like even more extreme examples of seed. So if you're given a plant and you look at a seed, you can never tell that the seed became a plant. But in experimental biology, we are limited by the endpoint measurements, and we try to conclude things which are not necessarily going to make an impact, right? So that's a very important point. So how do we address this problem of trying to connect state to a fate? We need some kind of a cellular time machine, and there are various ways to get at it. You can do longitudinal imaging, so you know which cell exactly became what. So Shaon's group does a lot of that, and other groups do a lot of that. Or there are other ways which will form my talk in 40 minutes. Is you can also trace cancer cells with barcoding approaches, which we develop and others have developed, and it's an active area of research. So which also leads me to sort of like conclude my talk by saying that there's a lot needs to be known, and as young scientists who are trying to get into cancer biology, there are experimental approaches, there are computational approaches, there are pure theory approaches. You can really pick your things that you like and get at these questions in very interesting and different ways. So with this, as I said, I'm here only all of tonight. I'm actually been in India for 2 weeks, so I really have to go back before my lab starts missing me. I hope they're missing me. But feel free to reach out to me over email, but I will stick around after dinner if people want to chat about anything.
Okay, I have one question. Can you go 2 slides back? Yeah. So me personally, when you talked about this kind of image, I've personally trained GRANs to do something similar. You have seen a lot of AI tools as of today that have come up on the market, even on Play Store, that you have Face app, which like converts from your baby pic from an adult pic or an adult pic from a baby pic. So I would have actually liked if you talked more about new machine learning approaches that have come up recently, maybe using graph neural networks or some other tools. Like I've personally been exposed to GANs, and I truly love.
Yeah, they're very effective. In fact, our lab does a lot of machine learning, and we try to predict it. And in many cases, we are very bad at it. So I
Yeah,
think
yeah, yeah.
machine learning is quite far. I think machine learning definitely, and our lab, 1/3 of the lab does a lot of machine learning on image data sets and try to
Yeah.
predict the future.
So even if you go back, something like fast RCNN or YOLO, kind of algorithms which do a lot of image processing. So can we use something along these lines in RNA-seq data? I don't think so. That
In what data? Sorry.
On RNA-seq data, I don't think so.
What is
RNA sequencing data?
RNA sequencing data.
Yeah, yeah.
Sure, if you know already that they're predictive, yes, but there's a lot still need to be learned, and I think AI definitely will play a big part in understanding cancer and predict behaviors, and it's already contributing, but it's not like one or the other. So they are all contributing in their own ways.
Other questions?
Yeah. Yeah, also, when I was getting my hands dirty with experiments for the first time, the most important feedback was take better images, right? And that's the start. So yeah, there's a lot of different things that are going on, and you just have to be open to different types of ways of thinking and technologies and mathematical frameworks. Maybe we can just break for coffee and people can chat over. 11:30. Yeah.